Hello, students. Welcome back again. In this lecture, we'll cover our last topic of our course, that is structure of deoxyribonucleic acid, that is DNA. So let's start. So this first we'll discuss about some basic general things about the DNA, and DNA is also a nucleic acid. It is a biopolymer, and this carries genetic informations for the development and functioning of an organism. Okay, like RNA, DNA does not exist as a single chain. Instead, two or more chains appear to interact with each other to with one another in the same way, and it forms like a helical structure. Okay, so the helical structure, or you can say double helix structure of DNA, was proposed by James Watson and Francis Crick in 1953, and the structure was primarily based on the model building by these two peoples and X-ray diffraction pattern of the molecule. And the combination of this X-ray diffraction pattern analysis with the chemical, you can say, modeling and their ideas, this so that the amount of adenine, that is the base, uh, was always equal to the amount of thymine base, and the amount of guanine base was always equal to the amount of cytosine base. So you can say that in this double helical structure of DNA, the number of adenine was equal to the number of thymine, and the number of guanine was equal to the number of cytosine base molecules. So these observations, they help them to conclude the structure of DNA, and that DNA consists of a polynucleotide chain wrapped around each other to form a helix. Okay, we'll see in the helical structure of DNA. Now, these bases like adenine and thymine, they are connected to each other by hydrogen bondings. Similarly, the bases guanine and cytosine, they are also connected with each other through hydrogen bondings. Okay, so hydrogen bonds between bases on opposite chains determine the alignment of the helix, and paired bases lie in planes perpendicular to the helix axis. Okay, now this sugar phosphate backbone is the outer part of that helix, and chain run in the anti-parallel direction. That is, if one chain goes from 3 dash to 5 dash, the other chain will come from 5 dash to 3 dash. They are alternative to each other. In anti-parallel manner, they are anti-parallel manner with each other. So you can see here, this is the double helical structure, or you can say the helix structure of DNA. Okay, so these rods type shape, you can see they are the backbone, which are composed of uh, sugar and you can say phosphate, and they are the bases. They are attached to the backbones and connected with each other through the hydrogen bonding. You can see the connections between them. Okay, so this is the helical structure. If one strand, let's say this strand is coming from 5 days, so the bottom end of this one will be 3 days, and if this is 5 days, it will be your 3 days end, because they have to be in anti-parallel manner. Okay, if for one strand 5 days is above, for other one the 5 days will be the downside. Okay, I hope you are able to understand. Now, the distance between these two portions, okay, the distance between these two portions, it is around 3.4 nanometer, and this diameter from here to here, this diameter is your 2 nanometer diameter. ठीक है, यहाँ से the distance between this helix from here to this helix up to here, this is your 2 nanometer. Okay, now this portion is known as major groove, the gap, and this, because this gap is very larger, this gap is very small, you can see it is minor groove. Clearly, diameter you can see here, this 2 nanometer. Now these bases, how they are bonded with each other. Also, I'll here you can see one portion. Let's say this portion is here. Okay, this is 5 dash end, and this is 3 end dash end of one strand. This is the 3 dash end of another strand, and the 5 dash end of the another strand. So here we can see the bases that are connected to each other alternatively. Let's say this is adenine. Okay, this is your cytosine base, this is thymine base, and this is guanine base. Now, if you see carefully, you will see the bonding between adenine and thymine These are bondings. Basically, these are the hydrogen bonds between these two. So adenine and thymine, they have two hydrogen bonds between them. So this adenine molecule is, you can say, attached to thymine by two hydrogen bonds. Similarly, if you look this structure, okay, so this is our, you can say, cytosine molecule, and this is our guanine molecule. So here we can see one, two, three hydrogen bonds between cytosine and guanine. So they are attached to, that is cytosine and guanine. They're connected to each other through three hydrogen bonds, and these hydrogen bonds are forming between the molecules, like adenine of one strand, thymine of the another strand, like this. They are not the both of the same strand. You can see this is our one strand, this is our the another strand. So this thymine attached to this strand and guanine attached to this left side strand. So the molecules are of different strand, or these bondings, actually these bondings, this is responsible for the helical structure of DNA. ठीक है? I hope you are understanding. So now I have a question for you here, guys, that will appear on your screen. You have to attempt that. Now, the denaturation and renaturation of DNA. So denaturation मतलब कि the double helical structures become single strand structure, and renaturation means कि double single strand strands they combine back to form a helical structure. So if we can say when we heat this DNA molecule in a solution, so these bonds between the, you can say the you can say uh, adenine and thymine and cytosine and guanine, they break down, and this double helical structure, it opens up in a single strand. You can see here is the double helical structure. You can see now when we heat it, so they become single strand structures. Okay, so this process is known as denaturation or melting of DNA by heating, converting into single strands, two different strands. Okay, now if we cool over this denaturated our DNA, okay, slowly if we cool it, it is the possibility to combine them again. That is to naturate again the DNA molecule. So you can see here these single strands DNA after renaturations, they combine back again to form renatured state of what? DNA. You can see this first and the last, they are the similar, and in between you have a denatured. Okay. So when heated DNA solutions cool slowly, the separate strands, they can combine, or you can say recombine, and forms the same base pair responsible for maintaining the double helix. Okay, I hope you are understanding what is denaturation and what is renaturation. Okay, now here again I have a question for you that will appear on your screen. You have to attempt that. Now, function of DNA. So DNA is the genetic material, therefore it is responsible for carrying all the hereditary informations. It has the property of replication, essential for passing genetic informations from one cell to the daughter cell, or from one generation to the next. That means the DNA transfer the property of one being to the another being. ठीक है? हम देखते हैं, हम मूवीज में भी देखते हैं, आजकल वैसे ही बहुत देखते हैं कि if you want to find the connection between two peoples, the best way, get their DNA test. If their DNA matches, there's a there is a fixed percentage. If that matches, so we can say that yes, these two persons, these two people, they are connected with each other. Okay, so we can do the DNA analysis of two persons, so we can find out the connection between them. Okay, now changes in sequence and number of nucleotides causes mutation that change in the structure of DNA, and this mutation, which is this is responsible for all variations and formations of new species. Okay, once we have some mutations or changing in the coding and decoding, or we can say the number of nucleotides in DNA, the DNA changes, and now the DNA will not match. Okay, again that depends upon how much nucleotides are number are changing, or how much not, how the sequencing is changing. Now the DNA controls all the metabolic reactions of cell through RNAs and RNA directed synthesis proteins, yani कि the RNA which are responsible for the protein synthesis. Okay, through them, this DNA has the ability to control the metabolic reactions. Okay, so I hope now you have an idea about DNA and its structure. So with this, we end up with our course. I hope you have enjoyed this course, and you have learned a lot of the things about nucleic acids, RNA, DNA, nucleoids, and nucleosides. Okay, so thank you so much for spending your time for my course. Thank you.